Stem cells and totipotency. In multicellular organisms, cells are specialised to perform specific functions. The process by which each cell develops into a specialised structure suited to the role that it will carry out is known as cell differentiation. Cell differentiation and specialisation. Single-celled organisms perform all essential life functions inside the boundaries of a single cell. Although they perform all functions adequately, they cannot be totally efficient at all of them, because each function requires a different type of cellular structure, enzymes, and other proteins. One activity may be best carried out by a long, thin cell, while another might suit a spherically shaped cell. No one cell can provide the best conditions for all functions. The cells of multicellular organisms are each adapted in different ways to perform a particular role. In early development, an organism is made up of a tiny ball of cells, all of which are identical. As it matures, each cell takes on its own individual characteristics that adapt it to the function that it will perform when it is mature. All the cells in an organism, such as a human, are derived by mitotic divisions of the fertilized egg, or zygote. It follows that they all contain exactly the same genes. Every cell is therefore capable of making everything that the body can produce. A cell in the lining of the small intestine has the gene coding for insulin, just as a beta cell of the pancreas has the gene coding for maltase. So why do the cells of the small intestine produce maltase rather than insulin, and beta cells produce insulin rather than maltase? The answer is that although all cells contain all genes, only certain cells express certain genes at any one time. Some genes are permanently expressed, which means they are switched on in all cells. For example, genes that code for essential chemicals, such as enzymes involved in respiration. These are expressed in all cells. Other genes permanently switched on in all cells include those coding for enzymes and other proteins involved in processes such as transcription, translation, membrane synthesis, ribosomes, and tRNA synthesis. Other genes permanently not expressed, or switched off, are genes for insulin in cells lining the small intestine. Further genes are switched on and off as and when they are needed. Differentiated cells differ from each other, often visibly so. This is mainly because they each produce different proteins. The proteins that a cell produces are coded for by the genes it possesses, or more accurately, by the genes that are expressed. Totipotency. An organism develops from a single fertilized egg. A fertilized egg clearly has the ability to give rise to all types of cells. Cells, such as fertilized eggs, which can mature into a body cell, are known as totipotent cells. The early cells that are derived from the fertilized eggs are also totipotent. These later differentiate and become specialized for a particular function. For example, mesophyll cells become specialized for photosynthesis, and muscle cells become specialized for contraction. This is because during the process of cell specialization, only some of the genes are expressed, and this means that only part of the DNA of a cell is translated into proteins. The cell therefore only makes these proteins that it requires to carry out its specialized function. These proteins include those required for essential processes like respiration and membrane synthesis. Although it is still capable of making all the other proteins, these are not needed, and so it would be wasteful to produce them. In order to conserve energy and resources, a variety of stimuli, controlling factors, ensure the genes for these proteins are not expressed. The ways in which genes are prevented from expressing themselves include preventing transcription and preventing translation. Stem cells. If specialized cells still retain all the genes of an organism, can they still develop into any other cell? The answer is, it depends. Xylem vessels, which transport water in plants, and red blood cells, which carry oxygen in animals, are so specialized that they lose their nuclei once they mature. As the nucleus contains all the genes, then clearly these cells cannot develop into other cells. In fact, specialization is irreversible in most animal cells. Once cells have matured and specialized, they can no longer develop into other cells. In mature mammals, only a few cells retain the ability to differentiate into other cells, and these are called stem cells. Stem cells originate from various sources in mammals. Embryonic stem cells come from embryos in early stages of development, and they can become any type of cell in the initial stages of development. Umbilical cord blood stem cells are derived from umbilical cord blood and are similar to adult stem cells. Placental stem cells are found in the placenta and develop into specific types of cells. Adult stem cells, despite their name, are found in the body of tissue of the fetus through to the adult. They are specific to a particular tissue or organ within which they produce the cells to maintain and repair tissues throughout an organism's life. Types of stem cells. Totipotent stem cells are found in the early embryo and can differentiate into any type of cell. Since all body cells are formed from a zygote, it follows the zygote is totipotent. As the zygote divides and matures, its cells develop into slightly more specialized cells called pluripotent stem cells. These are found in embryos and can differentiate into almost any type of cell. For example, embryonic stem cells and fetal stem cells are pluripotent. Multipotent stem cells are found in adults and can differentiate into a limited number of specialized cells. They usually develop into particular cell types. For example, stem cells in the bone marrow can produce any type of blood cell. Another example are umbilical cord blood stem cells. These are multipotent stem cells. Finally, unipotent stem cells. They can only differentiate into a single type of cell, and they are derived from multipotent stem cells and are made in adult tissue. For example, cardiomyocytes. Induced pluripotent stem cells, also known as iPS, are a type of pluripotent cell that is produced from unipotent stem cells. The unipotent cell may be almost any body cell. These body cells are then genetically altered in a lab to make them acquire the characteristics of embryonic stem cells, which can then become pluripotent cells. To make the unipotent cell acquire the new characteristics involves inducing genes and transcriptional factors within the cell to express themselves. In other words, to turn on genes that were otherwise turned off. The fact that these genes are capable of being reactivated shows that adult cells retain the same genetic information that was present in the embryo. iPS cells are very similar to embryonic stem cells in form and function. However, although they express some of the same genes that are usually expressed in embryonic stem cells, they are not exact duplicates of them. One feature of particular interest is that they are capable of self-renewal. This means they can potentially divide indefinitely to provide a limitless supply. As such, they could replace embryonic stem cells in medical research and treatment, and so overcome many of the ethical issues surrounding embryos. However, if they can keep on dividing, they can become cancerous and become a tumour. There are many possible uses of pluripotent cells. The cells can be used to regrow tissues that have been damaged in some way, for example by accident in the case of skin grafts for skin damaged by burns, or as a result of disease in the example of neurodegenerative diseases such as Parkinson's.